Mechanics of muscle contraction. Muscle fibers are long striated, cylindrical, multinucleated cells with many mitochondria. These cells have subunits called myofibrils, which are long organelles that fill almost the entire cell. Myofibrils are composed of repeated sarcomeres, each a contractile unit of muscle. The banding pattern of muscle fibers is caused by myofilaments within the sarcomeres. Thick filaments are composed of myosin, linear protein with cross bridges, and thin filaments of actin, which are globular protein. The contraction of a muscle fiber begins at the neuromuscular junction, where the presynaptic and postsynaptic membranes meet. The presynaptic membrane is a bilayer of terminal button, one of the many branches off of the end of the motor neuron's axon. The postsynaptic membrane is part of the sarcolemma, the muscle fiber's plasma membrane. At the places on the muscle membrane adjacent to the terminal buttons, there is a motor end plate where the sarcolemma is depressed and the membrane is thickened. The terminal buttons and motor end plates never touch, and the space between them is called the synaptic cleft. When the action potential and electrical impulse for contraction reaches the axon terminal, the calcium channel is open, releasing many calcium ions. This triggers vesicles within the neuron to carry neurotransmitters called acetylcholine to the presynaptic membrane and empty into the synaptic cleft. Acetylcholine is responsible for the contraction of all skeletal muscle. Once the acetylcholine exits the vesicle, it binds to nicotinic acetylcholine receptors on the sarcolemma and increases the conduction of sodium and potassium, causing a depolarization potential, which sends action potential in both directions along the fiber. This action potential will also spread into the fiber along the T-tubule system, made of tubules which fold in from the sarcolemma to cause muscle contraction. Depolarization spreads from the T-tubules membranes spanning the width of the cell to the sarcoplasmic reticulum, the smooth endoplasmic reticulum surrounding every myofibril like a sleeve, making it release large amounts of calcium ions which then flood the sarcomere. This calcium then binds to troponin, a protein on the actin filaments, and triggers it to move the tropomyosin protein, which blocks the myosin binding site on the actin. ATP is required for the motion of these proteins. The myosin cross bridges can then bind to the actin. Rigor mortis is the rough structure of muscles that is developed shortly after death due to the crossing between thick and thin filaments. Tetanic stimulation of the muscle results in the depletion of ATP. This provides no ATP left to break the actin-myosin cross bridges. This prevents myosin from binding to receptor sites on the thin filament, leaving the muscle in a flaccid state. The sliding filament theory of muscle contraction holds that, using ATP, the myosin then slides the actin filaments towards the center of the sarcomere through repeated attachments and detachments, similar to the motion of an inchworm. The contraction of many sarcomeres at once results in gross muscle movement. Other types of contractions include muscle twitches, rapid in response to a single stimulus periods, or phases of a twitch. Tetany, steady contraction of a muscle without distinct twitching. As said before, ATP is the direct energy source for muscle work. Muscle contraction can continue for a long time as long as there is adequate ATP. The supply of ATP depends on oxygen availability and organic sources. Three main sources of ATP generation is creatine phosphate glycolysis and oxidative phosphorylation. Creatine phosphate functions in the muscle cell by storing energy that will be transferred to ADP to resynthesize ATP. As creatine P runs out, the cell mobilizes its glycogen stores for the generation of ATP via glycolysis. Muscle tissue during this time must use the process of glycolysis to provide a source of energy until the cardiorespiratory system can supply enough oxygen to meet ATP standards aerobically. This happens, for example, when an individual sprints across a field and gains speed by exerting much force on the muscles in their body. The sprinter, by the end of the run, will have exhausted nearly all of his or her ATP supply and go into the process of glycolysis due to extreme oxygen debt. Because the majority of the ATP provided from the creatine P and the cardiorespiratory system becomes depleted, an anaerobic process known as lactic acid fermentation must be used to increase ATP and fulfill oxygen debt. This results in the production of lactic acid, which causes the proteins in the muscle to denature, or in other terms, muscle fatigue. As the action potential is occurring, the acetylcholine is being broken down by cholinesterase, an enzyme present in the synaptic cleft of synapses. This is why each nerve impulse should cause just one muscle contraction. After the impulse, a calcium pump is responsible for returning the calcium ion concentration to normal. Removal of calcium from the troponin means that the myosin head can no longer attach to the tropomyosin, and ATP is used to separate the myosin and actin filaments.